Hi, my name is Michael Johnson. I'm an associate professor here in the Department of Immunobiology at the College of Medicine Tucson at the University of Arizona. The focus of my research is looking at how metals impact microbes. Our bodies actually use copper to kill microbes that invade. We use it for metabolism, but bacteria don't use copper as much. In fact, copper is actually quite toxic to bacteria. So what my laboratory does is we try to maximize the copper's ability to kill the microbes so that we can find new therapeutics. Our bodies use metals, they use metals, and there's this constant competition between our bodies and the bacteria that enter us for metals that both of us need. However, there are metals that the bacteria does not need, in fact, are toxic to the bacteria, but to us are actually beneficial. One of those metals is copper. And what our technology does is it actually takes the copper and synergizes with it to force feed that to the bacteria in order to kill them. The problem in the marketplace is that antibiotic resistance is increasing and antimicrobial drug development is actually decreasing as far as being approved by the FDA. So what we wanted to do is come up with an antimicrobial that isn't a unitasker like most of the antimicrobials that are on the market today. Some antimicrobials will target a cell wall. They might target a ribosome. They might target this protein or that protein. What our technology does is it gives the bacteria copper, which can actually target a multitude of different pathways in that particular organism, making it very hard for it to adapt to that particular stress, which leads to bacterial death. When this project started, I really just wanted to understand how bacteria processed metal. That was really what I wanted to learn how to do. But then as we started working on it, I saw more and more relevance to being able to not just help target one particular pathway, but really go down the path of making a substantial therapeutic that we thought could actually help people. And lo and behold, we found a compound that synergized with copper to kill Streptococcus pneumoniae. So when we started looking at how this compound synergized with copper to kill this bacteria, we were absolutely floored. We said, in the test tube it worked, in the animal model it worked. We were excited. We were so excited. And then we said, well, what else can we try it against? We tried it against the thing that causes strep throat. We tried it against things that cause neonatal meningitis. We tried it against MRSA. And in every step of the way, our compound, our technology, worked with copper to kill these particular bacteria. My passion is in the laboratory, and I would really need someone who's as passionate about this particular technology, but on the business side of things. We know that these particular bacteria are on the CDC threat list. We know they all have these different antimicrobial phenotypes and things that really wreak havoc inside of what's going on with not just the US, but worldwide. So we're so excited that this particular technology can really be hopefully put into place to cure some of these infections that people actually have.